Benthic microfaunal community structure of the St. Lucia estuary mouth following the latest restoration project. St. Lucia is one of the most important estuarine systems in South Africa, but has a complicated history with mouth manipulation. Umfolozi and St. Lucia used to share a common mouth, with Umfolozi swampland effectively filtering sediment before entering St. Lucia. But in the 1950s, the swampland was canalized for agriculture and the two systems were artificially maintained separate to prevent the significant silt from entering St. Lucia. This separation deprived St. Lucia of primary source of fresh water, and devastating drought led to hypersalinity and habitat fragmentation, which threatened its integrity. In an effort to restore natural functioning, a global environmental facility-funded project relinked Umfolozi to St. Lucia. Benthic microfauna play a vital role as intermediate trophic level, linking primary producers to secondary trophic level, and they also respond differently to unstable environmental conditions. The aim of the study was to monitor the biotech component, including the microfaunal assemblage, before, during, and after the project activities. The mouth area was sampled quarterly between February 2015 and November 2018, with 6 sampling occasions prior to the project, 4 during, and 6 after the project activities. Salinity was significantly different during phases, with before being different from during the project. Sediment organic matter was also significantly different, with before being different from after. This was due to the freshwater input from Umfolozi that resulted in a significant reduction of salinity and increased SOM. There was a difference in mud contribution, with before being different from during and after. This was due to the silt from Umfolozi that showed an obvious threat to the St. Lucia system. There was no significant difference in abundance during the study period. Models in Table 1 shows that salinity had a significant relationship with species abundance, while temperature and salinity had a relationship with the presence and the absence of species. Phases were significantly different from each other, and NMDS plot in Figure 5 was used to visualize these differences. Overall deviance in Figure 6 shows that before was dominated by estuarine species, while during and after was dominated by freshwater species. Environmental stress often results in a decrease in abundance and diversity, and this supports the dominance of a single or a group of opportunistic species. This was evident in this study, as there was an increase in abundance of invasive species such as T. cranifera and opportunistic species such as H. cooperi. There was also a complete shift in groups from estuarine resident to freshwater taxa. This was due to reduced salinities. This project succeeded in increasing St. Lucia water level, and it will prevent habitat fragmentation and hypersalinity during dry conditions. Unfortunately, Umfolozi sediment is still a threat as it has traveled as far as South Lake, unless measures are taken to alleviate this sediment. Additional to the existing human impact on environment studies, this information may be used to advise St. Lucia conservation and management decisions. Thank you.